Looks like it's time to get started. Will you guys let me know you can hear me and see the PowerPoint I just had up? So if someone would just unmute or shoot me a text, let me know you can hear me.
We can hear you. It's good.
Very good. Thank you. All right. Well, today we're doing the second part of bioenergetics, which is going to be photosynthesis and cell respiration today. And we're going to start a little bit on cell respiration. We'll do a quick overview, and then we'll do some FRQs. That's the plan. So some important things about cell respiration. Cell respiration's job is to take the energy out of food, transfer that energy to the bonds of ADP to make ATP. It's kind of like charging up a battery. And then ATP is going to be the power source for pretty much everything our body does. Every living thing on Earth that we know of does some type of cell respiration. Now, there are two main types. There are aerobic and anaerobic versions. Every living thing does one or the other, or most things actually do both of those. We're also including plants, bacteria, fungi, every living thing on Earth. Down at the bottom, we see the equation for the aerobic version of cell respiration. And if you remember anything from intro chemistry class, I hope you see that reaction is a type of combustion reaction. Essentially, as your body does cell respiration, it's doing a chemical burn of sugar, reacting it with oxygen, getting the energy out, transferring that energy to the bonds of ADP to make ATP. That's the purpose. And then as waste products, some carbon dioxide, which we'll breathe out into the air, is released, and also some water vapor, which some of that we'll breathe out. Mostly we're going to recycle that water in our body, though. So the purpose is to take energy from food, make ATP. The waste products are carbon dioxide and water. The reactants are sugar, glucose typically, and oxygen gas. Every living thing on Earth does some type of cell respiration. As I said just a second ago, everything does it. It works basically the same way in all organisms. It's an example of a shared metabolic pathway between all of life. A set of reactions that involve the same enzymes, the same reactions in just about every living thing. And that's a good example of something that shows that all life has common descent. We're all related to each other. So we're going to focus on aerobic respiration first. Aerobic respiration, we tend to divide it into 3 steps. Glycolysis, the first step, which happens out in the cytosol or cytoplasm of every cell. Krebs Cycle, the second step, happens in the innermost part of the mitochondria, the matrix. Now that's in eukaryotes. We'll talk about prokaryotes a little bit later. And then the electron transport chain, which actually generates most of the ATP. This happens on the inner mitochondrial membrane on little folds called cristae. So glycolysis is the first step in both parts of respiration, aerobic and anaerobic. Excuse me. It's essentially the splitting of glucose and the removal of some of the energy of that glucose. Occurs in the cytoplasm. It makes 3 main products. It produces a little bit of ATP. Turns out it actually makes 4 ATPs, but it also requires 2 to happen. So you net 2 ATPs. Some of the energy from the glucose is also transferred to an electron carrier molecule called NADH. And then what's left of the sugar are 2 3-carbon pieces called pyruvic acids, or sometimes you'll hear those called pyruvate molecules. Most of the energy from the sugar is still in these pyruvate molecules at the end of glycolysis. Here's just kind of a summary. Here are the things you need to do glycolysis. Glucose, some NAD+, you need some ATP to act as activation energy. You also need some ADP and P to charge up. Here are the products. And again, NADH and ATP are both forms of stored energy that was originally in the glucose molecule. So step 2, it happens, like I said, in the mitochondria. That's the Krebs cycle. Step 3 also happens in the mitochondria. We'll talk about that. Every cell essentially in your body has mitochondria, but some cells are going to have more than others. The cells that are really active, like muscles, for example, are going to have lots and lots of mitochondria because they need lots of ATP. But cells like skin cells may not have as many because they're not doing a whole lot. Here's a picture of the mitochondria. Remember that we think that it started out as an aerobic bacteria that was phagocytized by another, probably a prokaryote, and that they eventually evolved through endosymbiosis to so that the smaller bacteria, the aerobic bacteria that was phagocytized, essentially became an organelle. In this case, mitochondria. Some evidence for that are the 2 membranes that we see. The outer membrane, nice and smooth, and then this inner one, it's all convoluted or curved. Scientists think that that membrane was probably the leftover remains of the original bacterial membrane. Notice the matrix is the area in purple, and the little folds on this inner membrane, these little folds, those are the cristae, and that's where the electron transport chain is going to take place. So sometimes we might actually think of aerobic respiration as 4 steps. In between glycolysis and the Krebs cycle, the 3-carbon pyruvates, remember those pyruvates that are left over are 3-carbon molecules. Before Krebs cycle can start, those have to be converted to 2-carbon molecules called acetyl groups. So in between glycolysis and Krebs cycle, some enzymes convert the 3-carbon molecules that are pyruvates into 2-carbon acetyl groups, and those are actually the molecules that enter into the Krebs cycle. That step is called the pyruvate conversion. Sometimes you'll hear it called the transition step. Happens in the mitochondria, typically in the matrix. It does convert the extra carbon here is released as carbon dioxide. So it's going to release some CO2. It's also going to move some energy from the pyruvate into the electron carriers, NAD plus or FAD plus, to make either NADH or FADH2, stored forms of energy that was originally in the sugar molecule. So Krebs cycle is essentially the oxidation of the pyruvates. Oxidation, remember, means removal of electrons, removal of energy. In this case, the energy is removed from the pyruvate and it's transferred mostly to these guys, NAD plus and FAD, to make NADH and FADH2, stored energy that's going to be used later to make ATP. Also during Krebs cycle, as the sugar is getting oxidized, the carbon molecules are released. So a lot of CO2 is released during this process. This is the CO2 that you're breathing out. CO2 that you're breathing out right now was originally sugar that you ate. Krebs cycle also generates a little ATP. That's per glucose. So all these numbers are per glucose that goes through the cycle. Main products though are NADH and FADH2. When Krebs cycle is over, most of the energy that was originally in the sugar, in the glucose, has now been transferred into these electron carriers. And that's going to lead us to step 3, which is the electron transport chain. So let's think of this as a mitochondrion. And remember that in the matrix here, this is where the NADH is. And we're going to only talk about the NADH. FADH would also have been generated in there as well. There are proteins embedded in the inner mitochondrial membrane in the cristae. Those are called cytochromes. They make up the ETC, the electron transport chain. What happens mostly during the electron transport chain is that this first cytochrome grabs onto an NADH and it oxidizes it. It takes away electrons, takes away energy. Let's just think about the electrons first. What's left of the NADH is NAD plus. These electrons then move across the cytochromes, across the ETC, and as they're moving, their energy is used to power the pumping of protons or hydrogen ions. And those H pluses get pumped from the matrix out here into this space between the two membranes out in here. And we start to build up a high concentration of H pluses out here. So active transport of H plus from the matrix out into what's called the intermembrane space. And what ends up happening as more and more H pluses get built up, we have a bigger and bigger gradient. Gradients, in this case, a proton gradient or an H plus gradient, are ways to store energy, potential energy, because those H pluses, since there's more on the outside than there is on the inside, they want to move in. But they can't just move in anywhere, because that inner membrane is made of phospholipids, and they're ions. Ions have a hard time crossing phospholipid membranes. But there is a way in, and that way in looks sort of like a little mushroom, and it's an enzyme and a channel protein. It's called ATP synthase. So H pluses will shoot through that ATP synthase with lots and lots of energy, lots of kinetic energy. And that enzyme will use that energy to put together ADP and P plus that energy from those moving H pluses to make ATP. This process of using an H plus gradient to make ATP is called chemiosmosis. And it's how most ATP is generated in your body. It happens this way in an electron transport chain, and something very similar happens in photosynthesis. So lots of ATP are generated here. This is where most of your ATP comes from in cell respiration. The one thing that we haven't mentioned here is the role of oxygen. We really don't need oxygen gas until the very last step of the electron transport chain. Oxygen gas will come to this last cytochrome and accept those electrons. It's called the final electron acceptor. And when it does, the oxygen gets reduced and it ends up combining with H pluses to make water. So this is where the water that's produced during cell respiration comes from. Without oxygen, the electrons on these cytochromes build up, and eventually they start repelling each other and the whole process shuts down. So why suffocation or drowning or something like that ends up killing you is because there's no oxygen, which means that the electrons build up on the ETCs, which means you can no longer generate very much ATP. And you essentially die from not so much a lack of oxygen, but from a lack of energy that you can use in the form of ATP. So that's aerobic respiration. Remember that chemiosmosis is the production of ATP using energy from a proton or an H plus gradient. You'll also sometimes hear this last step, this ETC step, referred to as oxidative phosphorylation. And what I want you to think about there is it's called oxidative because the first thing that needs to happen is the NADH and the FADH2 both get oxidized. The energy and electrons are taken from those. Their electrons and their energy are used to power the pumping of the H plus gradient. And the H plus gradient then provides the energy to phosphorylate ADP to make ATP. So that's why we'll sometimes hear this called oxidative phosphorylation. Oxidation of the electron carriers, where the energy from those electron carriers is used to phosphorylate ADP to make ATP. So here's just kind of a summary, and there's no need for you to know these numbers, but from each glucose, essentially, glycolysis produces 2 ATPs during glycolysis, 2 more net in Krebs cycle, somewhere around 30 to 32 in electron transport, for a total of 36, maybe 38 per glucose. That number varies a bit, and we still don't know exactly. It depends on conditions in your body, but somewhere around 36 ATPs per glucose. I want to take just a minute and talk about anaerobic respiration, and then we're going to get to an FRQ. So most organisms actually do anaerobic respiration. in addition to aerobic respiration. There are a few things, like some bacteria and some really, really small organisms, that only do anaerobic respiration. But there aren't very many of those things. The reason for it is anaerobic respiration is very, very inefficient. It doesn't make much ATP. We saw a second ago that aerobic respiration makes somewhere around 36 ATPs per glucose. Anaerobic respiration, on the other hand, only produces about 2. So it's not efficient at all. Starts off just like aerobic respiration does with glycolysis. Glycolysis works exactly the same way in anaerobic respiration as it does in aerobic respiration. It produces the pyruvate molecules, the NADHs, and the 2 net ATPs. So the 2 ATPs produced actually happen in glycolysis. Step 2, though, is fermentation. Fermentation's job is to take the NADHs that are generated in glycolysis and oxidize those back to NAD+. Because it turns out the cell only has a limited amount of NAD+, and without it, glycolysis can't happen. This step right here is not a problem when you're doing aerobic respiration, because that's one of the main things that happen during the electron transport chain, or oxidative phosphorylation. Fermentation, though, has to do it a different way. So fermentation, there's 2 types that we talk about in living things: alcoholic and lactic acid. In alcoholic fermentation, essentially what the cell does is it takes the pyruvate. It also takes the NADH generated from glycolysis, essentially reacts those together, produces NAD+, which is the purpose for fermentation. And as waste products, it generates some carbon dioxide and some ethanol. This happens in mostly yeast. In most animals, on the other hand, same kind of thing happens, except instead of generating CO2 and ethanol, animals and a lot of bacteria do this as well. They generate lactic acid, a waste product. This is the stuff that builds up in your muscles when you're really working out hard and you can't get enough oxygen to your muscles. We can't live with anaerobic respiration alone simply because it's so inefficient that it can't generate enough ATP to keep our cells alive. All right, I think that's where we're going to stop right now for cell respiration. So let's pull up our first FRQ. And I want you guys to take just a minute to read through it. And then we'll go through and try to answer it together. If you guys will take just a minute and read that, take a minute, look at the data table. We'll talk our way through the answers here in just a minute. All right, so we've got these corn plants, corn seedlings of equal size. We're assigning them to one of 3 groups randomly. Group 1, we just dried those to figure out essentially what the mass of the initial plants were, and that mass was 14.8. 10 more plants were put in a group that was exposed to light. 10 more were put in a group exposed to dark. All the conditions other than the light or dark were the same. So they were in the same pots. They were the same kind of plants. They got the same amount of water. Those were experimental constants. At the end of the week, the plants in groups 2 and 3 were dried, and the mass was redetermined, remeasured. And we see that in group 2, the plants that were in the light actually gained mass. They gained about 18 grams of mass. Whereas the plants in group 3, the ones that were in the dark, they actually lost mass. It looks like they lost about 3.1 grams. So first question, to explain the increase in mass of light-grown plants, look at that first verb, identify one inorganic source of new plant mass and connect it to the cell process underlying the increase in mass. So 2 important verbs, identify, connect. Also, inorganic source of new plant mass. So an inorganic form. What I'm hoping that you realize is that there's really 2 answers you could put here, 2 easy answers. And those 2 answers are carbon dioxide, which is considered to be an inorganic form of carbon, and water. So first of all, I would say that one form of inorganic, one inorganic source of new plant mass is carbon dioxide, or one inorganic source of new plant mass is water. Only answer one or the other. Now we've got to connect it to the cell process underlying the increase. Well, I'm hoping you realize that if plants are in the light, what's making them gain mass is that they're doing photosynthesis. So you could say carbon dioxide is an important reactant for the process of photosynthesis. And this is as plants do photosynthesis, they make macromolecules and they gain mass. Same for water. Water is an important reactant for photosynthesis. Plants take in water. They use that water to make organic macromolecules, which causes them to increase in mass. Similar kind of thing is going on in part B. So part B says, to explain the decrease in the mass of the dark-grown plants, identify the overall chemical reaction that is occurring in the plant cells and connect it to the cell process underlying the decrease in mass. Well, they're in the dark, which means they can't do cell respiration. But what I'm hoping you realize is that every living thing has to do, they're in the dark, so they can't do photosynthesis. Let me go back and fix that. Every living thing has to do cell respiration. They have to do cell respiration to make ATP, which powers everything they're doing. So even in the dark, they're doing cell respiration. So we're going to identify the overall chemical reaction. You might get by here with saying the chemical reaction is cell respiration. I'm going to go ahead and though say the chemical reaction is aerobic cell respiration, and here's the equation for it. And we'll look at the grading rubric in a minute and see what it says. So that's our equation for aerobic cell respiration. That's the equation. So I would say that as we've got to do the connect part of this. So the connection is as plants do cell respiration, they're breaking down the carbon dioxide and releasing, let me say that again, they're breaking down the sugar and they're releasing carbon dioxide. This carbon dioxide is coming out of the plants. It's coming out through the stomates, the little holes on the bottoms of the leaves. So they're losing mass because they're losing carbon dioxide. They may also lose a little water, but they're probably bringing in as much water as they're losing, more than likely. So let's go to the grading rubric and see what it says for us. Grading rubric on this one is pretty simple. So part A, remember we were trying to explain why the plant increased in mass. So you could have got a point for identifying either carbon dioxide or water. And essentially you got another point for saying that either the carbon dioxide or the water are incorporated into carbohydrates by photosynthesis. You probably didn't even have to say carbohydrates. You could have said sugars. You could have said organic molecules. I think they would have taken a lot of things on part A. For part B, so we're trying to explain why the plants lost the water. So they did want us to give the actual equation, and you could do that in actual symbols, or you could do it in words. And then they wanted us to essentially say that During cell respiration, the plant releases carbon dioxide into the environment, and this is why it's losing mass. So 1 point for the identification of the equation, and 1 point for the connection that the mass is lost because CO2 is given off during cell respiration. All right, before I go further, questions on that FRQ? Or cell respiration in general, for that matter. All right, well, here's one more cell respiration FRQ. So take a minute and read through that one. Go ahead and read parts A, B, and C. Okay, so this enzyme, PDC, catalyzes the conversion of pyruvate, remember that 3-carbon molecule, to the acetyl molecule, the 2-carbon molecule, a substrate for the Krebs cycle. The rate of pyruvate conversion is greatly reduced in individuals with this PDC deficiency, a rare genetic disorder. Identify the cell location where PDC is most active, and then we want to make a claim about PDC deficiency. So before we try any of that, reminder of some stuff. Remember that in aerobic cell respiration, we have glycolysis. That's step 1. That happens in the cytoplasm. Step 2 is generally considered to be the Krebs cycle, which happens in the mitochondrial matrix. But remember, in between glycolysis and Krebs cycle, there's the step we're talking about up here, which is the pyruvate conversion. This also happens in the mitochondria, mainly in the matrix. And it is when 3-carbon pyruvate is oxidized down and converted to a 2-carbon acetyl group, which is what this question is asking us about. So this is the step we're talking about. Remember that step 3 is the electron transport chain, or sometimes referred to as oxidative phosphorylation. So part A, identify the cellular location where PDC is active. Well, this conversion happens in the mitochondrial matrix. So I think you could have got by on part A by simply saying this process for PDC is most active in the mitochondria or in the mitochondrial matrix. Either way, you could have gotten a point on that. Part B, make a claim about how PDC deficiency affects the amount of NADH produced by glycolysis and the amount produced by Krebs cycle. Provide reasoning to support your claim based on the position of PDC catalyzed reaction, blah, blah, blah. So essentially, notice that the pyruvate conversion happens after glycolysis. So I'm hoping that you can see that your claim about glycolysis should be that this deficiency should have no effect on the amount of NADH produced by glycolysis, because the pyruvate conversion happens after glycolysis has already occurred. So this deficiency should have no effect on the amount of NADH produced by glycolysis. Now, on the other hand, notice that the pyruvate conversion happens before the Krebs cycle. And the 2-carbon acetyl group created by this conversion is actually the beginning reactant for Krebs cycle. So essentially, this deficiency should essentially stop the mitochondrium from making any NADH during Krebs cycle, because the acetyl groups are the reactants needed to start the Krebs cycle. Krebs cycle happens after the pyruvate conversion. Therefore, this deficiency is going to essentially limit or prevent the Krebs cycle from making any NADH. That's what they wanted you to talk about in part B. Now in part C, let's do some erasing here so we have some space. This is more of a genetics review on this part. And it says PDC deficiency is caused by mutations in the PDHA1 gene, which is on the X chromosome. So it's linked to the X chromosome. A male with this deficiency, so here, when we're dealing with sex-linked traits, we need to show sex chromosomes. So here's the male with the deficiency. It doesn't tell us if this is a dominant or a recessive kind of thing. I'm just going to put an asterisk here right now to let us know he's got the gene on his X chromosome. And so he has a child with a female who's homozygous with no family history. So she doesn't have this gene. So there's no asterisk on either one of her X's. And we want to calculate the probability that a male offspring will have the deficiency. Well, remember that what makes a male is that he gets the Y chromosome from dad and one of these X's from mom, either this X or this X, one or the other, to make him be XY. Since his X comes from mom, he cannot get this disorder because the only family member that has it is the father. So your answer here is 0. There's 0 probability that this couple will produce a female that has the trait. Now, if the trait happens to be a dominant trait, there is a chance that a female could inherit it because she gets one X from dad and one of these 2 X's from mom. So if it was a dominant trait, she could end up with that kind of genotype, and she could be affected by the trait. All right, that's it for FRQ2. I want to quickly talk just a minute. Before I do, questions on this FRQ? Here's the grading rubric. So the cell location where it happens, 1 point for mitochondria or mitochondrial matrix. For part B, 1 point for the claim that no change happened to glycolysis, the amount of NADH made by glycolysis. And another point for glycolysis continues, PDC is not needed, or glycolysis occurs before the conversion. An additional point for saying that the amount of NADH made during Krebs cycle decreases, and an additional point for explaining why. That Krebs cycle happens after the conversion, or that the acetyl groups are needed by Krebs cycle. And then finally, on part C, 1 point for essentially saying 0 probability or the offspring cannot have the deficiency. All right, I don't have a terrible amount of time left, but I'm going to quickly talk through photosynthesis. So here's an equation for photosynthesis. So plants take in sunlight. They need carbon dioxide. They need water. They're doing this reaction to make carbohydrates, which we're usually going to write out as glucose. And as a waste product, they release some oxygen gas, some of which leaves the plants through the stomata. So that's our overall equation for photosynthesis. Reactants, sunlight, carbon dioxide, and water. Products, sugar, carbohydrate, or oxygen gas and oxygen gas. As far as organisms that can do photosynthesis, we're talking about some bacteria, particularly the ones we might call cyanobacteria. We're talking about algae, some of the protists, like Volvox, for example, and we're talking about plants, all plants. So those are the organisms that can do photosynthesis. And remember, they not only do photosynthesis to make the sugar, they also do cell respiration to break down that sugar and create ATP with it. So we typically think of photosynthesis happening in 2 steps. And those 2 steps are the light-dependent reactions. and the Calvin cycle. The light-dependent reactions happen on these little membranes inside the chloroplast, which are inside the leaves. These are called thylakoids. Embedded in the membranes of these thylakoids are the photosynthetic pigments like chlorophyll. Also some ATP synthase. The Calvin cycle is the step that actually makes the carbohydrate. It happens out in this liquidy inner portion of the chloroplast called the stroma. So this is the light-dependent part of photosynthesis. So this is a vast oversimplification, but essentially these photosystems are groups of pigments. And these, think of them almost like solar panels. So light shines down, and these photosystems absorb light. As they do, the electrons in some of those photosystem molecules start to vibrate very quickly, and eventually they gain so much energy that they eject electrons. We usually think about it in terms of pairs. Both photosystems eject a pair of electrons. The electrons that leave photosystem 2 go across a carrier or a system of cytochromes in ETC, and they move from photosystem 2 over to photosystem 1. As they're moving, though, the energy of those moving electrons is used to pump H pluses, just like we talked about in photosynthesis. But in this case, the H pluses are pumped from the stroma into this inner part of the thylakoid, the inner thylakoid space. Again, we're building up an H plus gradient. Lots of H pluses in the thylakoid, a much lower concentration on the outside. The electrons that leave photosystem 1, they end up reacting out in the stroma with another electron carrier, NAD+, to make NADPH. Very much like the NADH that was made during cell respiration. This is a form of stored energy. Energy that originally, in this case, came from the sun. This NADPH will be used during the Calvin cycle as an energy source for making sugar. The other thing that happens here, though, is these H pluses build up and the concentration gets higher and higher inside the thylakoid. There's really only one way out because of the phospholipid membrane that makes up the thylakoid, and that's out through ATP synthase. These things shoot out with lots of kinetic energy, and that energy is harnessed by this enzyme, ATP synthase, to take ADP and P to make ATP. Again, this is the process of chemiosmosis. Using the energy from an H plus gradient, a proton gradient, to phosphorylate ADP to make ATP. Another source of stored energy. Energy that used to be sun energy, sunlight. So the first part of photosynthesis essentially makes 3 main products. We've only talked about 2 of them. So far, we've talked about the ATP and the NADPH. The other thing we should mention is that photosystem 2 lost electrons. Needs to replace those. It actually has a huge pull on electrons. And what it ends up doing is it grabs onto water molecules and rips those water molecules apart and essentially takes electrons from the water molecules. What ends up being generated during that process, which is called photolysis, is that what's left over from the broken down water are some H pluses, which contribute to this gradient in the stroma, but also oxygen. So the oxygen that we're breathing right now is released by plants, and they created this oxygen by ripping apart water molecules. So the 3 products of the light-dependent part of photosynthesis, oxygen gas, which is released mostly by the plant. They use a little bit of it to do cell respiration. And then the 2 forms of stored energy, NADPH and ATP. While we're here, and we'll see this in a minute in our FRQ, plants, this is what we call an absorption spectrum. It shows you what plants absorb best, what certain pigments absorb best. And plants' 2 main pigments are chlorophyll A and B. And you can see that these 2 pigments have peaks right in here. So they're both really good at absorbing bluish violet. They have another peak over here in the orange red area. So they're both pretty good at absorbing those as well. Better at absorbing blue violet, second best at absorbing orange red, But here in the middle, and the greens and the yellows, they pretty much reflect that. And this is why what we see from most plants, they appear green, because what we see when we look at an object is what's reflecting off of that object. Plants reflect greenish yellow light, therefore most plants appear green. They're absorbing the red, they're absorbing the blue and the violet. They're using those forms of energy to do photosynthesis. They can't really use the greens and the blues, so they reflect those. Let's go ahead and talk about the second part of photosynthesis, the Calvin cycle. So here's a simplified diagram of how the Calvin cycle works. Remember, this happens in the stroma of the thylakoid, and essentially the plant takes in carbon dioxide. This comes in through the stomata on the bottoms of the leaves, the little holes on the bottoms of the leaves. The carbon dioxide is essentially the building block of sugar. Once inside the leaf, an enzyme called rubisco will grab onto that carbon dioxide and attach it to a molecule called ribulose bisphosphate. This makes a pretty unstable 6-carbon molecule, which breaks down very quickly into 2 3-carbon molecules. Now notice we're starting here with 3 CO2s. So each one of those makes one of these short-lived intermediates, and those are going to all split in half. So we end up with 6 molecules, 6 3-carbon molecules, and I'm going to call these PGA molecules. 3-phosphoglycerates is the actual name, PGA. But I want you to think of this PGA sort of like half of a glucose without very much energy. So the next step that happens in the Krebs cycle is that the ATP that was generated in part 1, the light-dependent reactions, is used to phosphorylate the PGA. Give it phosphate groups, transfer energy to it. Notice the name changes once it gets phosphorylated. And then the other energy source that was made in part 1, NADPH, is used to reduce. Give it electrons, give it energy to the 1,3-bisphosphoglycerate. So all of this step from here to here, this is called reduction. This is putting the energy into the carbon dioxides, energy that originally came from the light-dependent part of photosynthesis. Once all that's said and done, we end up with molecules called G3P, glyceraldehyde 3-phosphate. I want you to think of this as a 3-carbon, essentially half of a glucose, but now it has energy. For every turn of the Calvin cycle makes 6 of these G3Ps. Well, it takes out one of those that's then used to make sugar or some other kind of organic molecule that the plant might need. The other 5, though, are carry on to do step 3, which is essentially needed to keep the cycle going. They're used to remake this guy, ribulose bisphosphate. Because if the plant runs out of this, the whole process shuts down. So this seems very inefficient, that it would do the cycle, make 6 of these G3Ps, but essentially only use one of the 6 to make sugar. The other 5 are used to just keep the cycle going. So it's not overly efficient, but this is the way plants evolved, and it works. And plants have become very, very successful because it does work. All right, I think that's as far as we're going to go with photosynthesis. Let's go ahead and look at the last FRQ, and then we'll call it a day. All right, so here's our last FRQ. If you guys will take just a minute and read that, you're going to need to see those graphs. I tell you what, for purposes of our time, let's just look at parts A and B for right now. So look at the graphs, look at the chart, read the intro little blurb there in part A and B. All right, so you can see in part B, we've got two verbs. Identify the pigment, chlorophyll A or bacteriorhodopsin, used to generate the absorption spectrum in each of the graphs above. Explain and justify your answer. So let's start with this one. Let's start with graph 2. And what I'm hoping that you see with graph 2 is that the pigment here absorbs really well right in here and absorbs really well right in here. Well, if we go down at this chart and we look at about 400 to 450, well, that's violet, bluish violet. And if we look around 650, 700, that's reddish. So red and violet. So those are absorbed well. And then if we look in here, somewhere between 5 and 600, 450 and 600, something like that, those are the greens and the yellows. So this plant is not absorbing green, this pigment, but it is absorbing red and violet. So I'm hoping that you see that graph 2 is chlorophyll. And the explanation and justification would be that chlorophyll absorbs violet, absorbs red, reflects green, and because it reflects green, that's why chlorophyll appears green to our eyes. That's what they wanted you to talk about on that part of part A. Now, graph 2, or graph 1 rather, is bacteriorhodopsin. And notice that it's peaking out around 550, 575, something like that. That's where it absorbs. So it's absorbing in the yellows and the greens. But notice over here, it's not absorbing much at all. That's 400 or so. That's the bluish violets. And over here at red, this 700 is red. So it's reflecting red and blue, but absorbing green and yellow. So we might say that graph 1 is bacteriorhodopsin because it's reflecting blues and reds, which combine to give it a purple appearance. And it's absorbing greens and yellows and using those as the energy sources for photosynthesis. That's how you got the points on part A. Now part B says in an experiment, identical organisms containing the pigment in graph 2, so they have chlorophyll, are separated into 3 groups. The organisms in each group are illuminated with light of a single wavelength, 650 for first group, 550 for the second group, and 430 for the third group. The 3 light sources are of equal intensity, and all organisms are illuminated for equal lengths of time. Predict the relative rate of photosynthesis in each of the 3 groups and justify your predictions. So if we look, group 1 is exposed to 650 nanometers, which 650 is reddish. It's right in here. So group 1 is given a red light. Group 2 is exposed to 550. Well, 550 is right in there, green light. And finally, group 3 is exposed to light that's at, where'd that go? 430 nanometers, which is right in here. That's violet. And we want to essentially know who does photosynthesis the fastest is essentially what the question's asking us. So if we look, because we know this plant has chlorophyll, it's best at absorbing around 430. It's second best at absorbing around the red, 650. And it's really terrible at absorbing in here. So I'm hoping that you see that group 3 is going to do photosynthesis the fastest because this pigment is best at absorbing violet light, and therefore it will have the most energy to do photosynthesis. Group 1 is going to do photosynthesis the second fastest because this pigment is second best at absorbing reddish light, and therefore more energy for photosynthesis. Group 2 is going to do almost no photosynthesis because these plants reflect green light and therefore don't have an energy source for photosynthesis. Here's the grading rubric. So let's blow this up a bit. So for part 1, 1 point for getting the pigments right. graph 1 was bacteriorhodopsin, graph 2 was chlorophyll A. And then another point for explaining that bacteriorhodopsin is purple because it's reflecting the blues and the reds and absorbing the greens. And then another point for explaining that chlorophyll A is green because it's reflecting greens but absorbing reds and blues. And then on part B, essentially saying what we said before, that the group with the highest rate of photosynthesis is the group exposed, group 3, to the bluish violet light. They get the greatest amount of energy because they're absorbing more of that color of light. Group 1 is the intermediate, the second best, the second fastest at doing photosynthesis because the plants are pretty good at absorbing the reddish colors of light. And then group 2 rather, is doing almost no photosynthesis because they're reflecting the green light. All right, that's it for today's webinar. Are there questions I can answer for any of you guys?